today's session we are going to study about a topic under the +1, that is health and diseases. And in this, one of the important topic is the immune system. So as you all of know that every day we are being get exposed to the various kinds of the infectious agents, but all of them are not responsible for causing of the diseases. And the reason for this is our body has got a self-defensive mechanism is present. And so it is the ability of the organism of the body able to defend against the various types of the disease causing agents or the infectious agents, and is called as the immunity. And so function of the immune system, we call it as immunity. And the basic requirement of the immune system is to detect the difference between the self and non-self. And so the ability of an individual to fight against the disease causing or the infectious agents and is called immunity. And this immunity is a part of the function of the immune system, which consists of various types of the cells, organs and the tissues, and the various types of the proteins that are secreted by the cells. And so all these are constitute to form the immune system. And so as we discussed, and that the basic requirement of the immune system is to differentiate between the self and a non-self, and to protect the body from the harmful organisms which are responsible for causing of the diseases. And not only that, the toxic substances that are released by these microorganisms are the cancerous cells that are get transformed inside the body. Against them also our body used to offer the protection. And so study of the immunity or the immune system, what we call it as immunology. And the father of the immunity is the Edward Jenner. Yeah, immune system is a bodyguard, is with a powerful arsenal, and that is provided with all the kinds of the cells and their secretions and the proteins all are together, which are can able to defend against this infectious agent. So we call it as a powerful arsenal. Now, to understand the immunology better, we need the certain basic concepts such as the lines of the defense or the cells of the immune system, organs that are involved inside the immune system, and the various kinds of the soluble mediators and immune system antigens, and all these are required to understand these basic concepts of the immunology. Among them, the first one is actually the lines of the defense. And our body actually provided with three lines of the defense are presented: the first line and the second line defense and the third line defense. And first and second line defenses are very common to all the living organisms. And whereas the third line defense is actually developed in response to the exposure to the in response to the microorganisms or the infectious agents, or we can say that the antigens. And innate immunity is another kind of the one defense that actually comes under this, and the defense that is developed after the birth, we actually call it as acquired immunity. And among them, there are the various kinds of the cells are also present, the cells of the immune system, and they are the lymphocytes are present, phagocytes are present, and also the auxiliary cells are also present. And out of this, the first we'll look at into the three lines of the defense, and the first line, second line, and the third line defense. And first line defense is actually offered by the in our body, the physical barriers are present. So especially the physical barriers that are present in our body are, the first one is of course the whole skin. Then another one is the mucous membrane that is present especially in the gastrointestinal tract, respiratory tract, urinogenital tracts. In all of them, the mucous membranes are present. And another one is the lysozyme that is present inside the tears and the saliva, and the sweat and the low pH of the sebum secreted from the sebaceous glands, and the mucus secretions of the gut, bronchi, bronchioles, and all they are which are responsible for the trapping of this dust or the microbes or the infectious agents. And all of them collectively functions as comes under the first line defense. In addition to that, the anterior microbial proteins produced by the epithelial layers, which also responsible for the preventing the entry of microbes. And so all this comes under the first line defense. And now let's move on to the second line defense. And so once the body could able to penetrate penetrate through the skin and cross all the mucous barriers and all, then it got exposed to the enter inside the body, and they are immediately get attacked by the phagocytic cells present inside the body, like the neutrophils or the monocytes. In addition to that, the NK cells and natural killer cells and the inflammation that is caused by the body in response to the infection, and the high temperature that is developed, and this inflammation and rise of the temperature, they all are also responsible for the preventing the rapid multiplication of the microbes. And so these two comes under actually the first and the second line defense, and it is actually non-specific, and say they can able to show the defense against the any kind of the microbes, and they are also present by birth inside the organism. So they become the part, these two lines of the defense become the part and parcel of the innate kind of the immunity. And coming to the third line of the defense, in case of that one, it is actually offered by the T cells and the B cells, and which are in addition to that the antibodies that are produced from the B cells, and out of these all the three lines of the defense, the whatever the defense that is offered by these the antibodies and all, it's the most powerful defense, and it is very specific. So on a particular type of an antigen to which our body get exposes, and against to that only the third line defense actually shows the defense. So this actually comes under the part and parcel of adaptive type of immunity or the acquired type of immunity, and it is also very specific lines of the defense. So like this, our body has got the three lines of the defense are presented. So failure of all the three types of the defenses, that is the first line defense, second line defense, and the third line defense. So actually the failure of all these three lines of the defense is actually responsible for the cause of diseases. So finally, if a body is suffering from the disease, or result of the disease in body is mainly due to the failure of all these three kinds of the defenses. So these are the one concept, and then let's move on to the different types of the cells that are present in case of the immune system. So there are the three kinds of the cells are present in case of the immune system, and those three types of the cells are, one is the lymphocytes, and the second one is the phagocytes, and the auxiliary are the additional cells that are present inside the body. So first move on to the lymphocytes. So lymphocytes, they are the chief cells of the immune system, and there are actually the three different types of the lymphocytes are present, and that is the B lymphocytes, T lymphocytes, and the NK cells, which are, and also the large granular lymphocytes called as LGLs. The first one among them is the B lymphocyte cells. And as you all know that B lymphocyte cells are actually get produced from the stem cells of the bone marrow, and in the adult cells, and also from the liver in the fetus stage. And in case of the birds, these B lymphocytes are actually get undergo this maturation or the production from the bursa fabriceus in case of the birds. So in the primary lymphoid organs, in those cells, these B lymphocyte cells undergo the maturation. See, lymphocyte is actually produced from the bone marrow only, but their site of the maturation is also in the bone marrow in case of the mammals, whereas in case of the birds, their maturation is at the bursa of fabriceus. So the only difference that exists, as you already studied the characters of the lymphocyte cells are actually smaller with a larger nucleus is present inside with a scanty cytoplasm. These are actually the normal lymphocyte characters. But the difference exists in between a mature lymphocyte that we call, what we call it as B lymphocyte. It is provided with the nucleus. The only thing is that it is provided with the membrane bound antibodies are present. So presence or acquiring of these membrane bound antibodies makes the cell to get maturation. Now we can call it as a B cell. So earlier it is a lymphocyte, the smaller cells with a larger nucleus and a scanty cytoplasm, but the difference exists in between lymphocyte and the B lymphocyte is getting exposure to these membrane bound antibodies, and now it becomes immunocompetent cell, and B lymphocyte cell, what we call it now, are the mature B cells we call it as, so that it can able to recognize a freely moving antigen inside the freely circulating antigens can be recognized with the help of these membrane bound antibodies, as they are provided with, here the antibodies are provided with the BCRN. B cell receptor, that what we call it as, which is present at these membrane bound antibodies. With the help of these BCRs, the B lymphocyte can able to recognize the freely circulating antigens. So once it recognizes this antigen or comes in contact with the surface of any kind of the antigen, Now the B lymphocyte cells become the functional cells, and now they immediately get to differentiate into two different kinds of the cells. And so after the coming encountered with that of the antigen or the infectious agent, now the B lymphocyte cells differentiate into two different kinds of the cells, and so they can take the antigens, and so the mature B cells, mature B lymphocyte cells, they are differentiate into the two different kinds of the cells, and they are the long-lived memory cells are like that, they get to differentiate, and the second type is the plasma cells, which are the effector cells. And so memory cells and plasma cells, like that they actually get differentiated. Of course, the memory cells are provided with these membrane bound antibodies are present, and the plasma cell, which is actually responsible for the production of the antibodies. And so these plasma cells, which we call them as effector cells, and memory cells, they remain and they have got the life for a long lifespan is present. And so these memory cells are very much specific to only one particular type of the antigen only, and so whatever the antigen to which this mature B cells comes to the exposure, to that same type of the antigen only the memory cells are specific. And so in that way, these memory cells are responsible for the production of the antibodies whenever the body comes to the exposure or the second time exposure of the same antigen, then these memory cells are responsible for the production of the further antibodies. And so the interaction between the antigen and this BCR of the mature B lymphocyte cell is actually brought about inside the secondary lymphoid organs. And so inside them actually they comes in contact with them. So if you look at the slide diagram, this is a B lymphocyte cell, B cell with a membrane bound antibody is present. Whenever it comes in contact with the antigen with the help of the BCR that is present, so the B lymphocyte cell becomes the activated end. So and later as an interaction of the secondary lymphocyte cell, the B lymphocyte cell in turn differentiates into memory cells and plasma cells responsible for production of antibodies. This only occurs only with the help of this T lymphocyte cells that we study in course of this session. We keep on going to study about that one. Now so the specific antigen and the memory cells are in turn, as we discussed earlier, the memory cells are in turn responsible for the production of the antibodies in a very quick manner as a result of the second exposure of the same antigen. And now let's move on to the second kind of the cells, and the second kind of the lymphocyte cells are the T lymphocyte cells. And now T lymphocyte cells cannot able to produce the antibodies, or they cannot able to recognize the freely circulating antigens, and so neither produce the antibodies nor recognize the free or circulating antigens. And so these are also actually get derived from the stem cells of bone marrow, and whereas after their production from the bone marrow, they get move on to that through the blood circulation, while they get passed through the thymus gland like the primary lymphoid organ, where they get differentiate into the T lymphocyte cells. And now they get transformed into the T lymphocyte cell. They are called as mature cells now, or the immunocompetent cells. And now the difference between that the lymphocyte and the T lymphocyte is only that one. And now this get transforms into the T lymphocyte cell, which is get provided with the TCR. And now we call it as TCR. And now this is called as a T cell. And so a normal T lymphocyte cell and it's a lymphocyte cell which get transformed into T cell. That means now it is get provided with the exposure of the TCR. Now it can able to recognize the antigen, but it cannot able to recognize the freely circulating antigen unless it is presented by an antigen presenting cell or the altered cells. And so T cell cannot able to recognize the antigen on its own, and unless it is get presented by the APCs or the altered cells or antigen presenting cells with the help of their Mhc molecules. And we'll study that what are these Mhc molecules and all in course. And so antigen presenting cells, as you know that there are the different kinds of antigen presenting cells are present. They are like the dendritic cells are present, macrophages are present, and the B lymphocyte cells. They are effectively function as antigen presenting cells. And let's move on to the different types of T lymphocyte cells. And T lymphocyte cells are actually classified based on the certain types of the glycoproteins and present on the surface of these T cells, and they are called cluster differentiation molecules. And so cluster differentiation molecules called the CDs, and they are the glycoproteins which are present on the surface of the T cells. And so based on that, they are get classified into two different types, and these CD markers are of two types, and they are the CD4 and the CD8. And based whoever the cells which are provided with a CD4 are called CD4 plus cells. The T cells which are provided with a CD8 marker are called as CD8 plus cells. And so they are nothing but T4 cells or T8 cells or TH cells are at the TH cells and are the TC cells, and which we call it as an. Among them, the first one is the T helper cells. And so T helper cells are provided with a CD4 marker. If you look at this one, so these are the T lymphocyte cell which is provided with a TCR. So with the help of that, it helps in the recognition of antigen, and this is a CD8 4 marker. So now we can call these cells as TH cells. So those cells that are provided with a CD4 marker, so those cells are called as CD4 plus cells or T4 cells. Now the another kind of the cells that we have got is those which are provided with a CD8 marker, and those are called as TC cells or CD8 plus cells or simply T8 cells. So CD8 plus cells. So TC cells are provided with a CD8 marker, TH cells are provided with a CD4 marker. They are the cluster differentiation molecules, which are nothing but a different kind of the glycoproteins that are present on the surface of the cell membrane. Now TH cells are the first kind of the cells are the TH cells, and these TH cells are actually involved in the both the kinds of the immunological responses, and that is they are both involved in the humoral immunity as well as cell mediated immunity. So without the help of these TH cells, the immune system almost get impaired, and because the humoral and cell mediated immunities are under the control of these TH cells. And so TH cells are the cells which are actually helps the B lymphocyte cells to produce antibodies, and they also helps in mononuclear phagocyte cells to perform the phagocytosis phenomenon, and they are also responsible for the activation of the TC cells to show the immune response called cell mediated immunity. So they make the B lymphocyte cells to produce antibodies, they make the phagocytic cells to show the phagocytosis, they make the TC cells to show the response called cell mediated immunity. So all these are under the control or with the help of these cells. That's why they are named as T helper cells. And these cells are also involved in the resections reactions, especially whenever the cells or tissues are transplanted. So and those are also resected with the help of these TH cells. And these TH cells are also responsible for the secretion of one kind of the cytokines called gamma interferons, and these gamma interferons stimulate the macrophages, and the macrophages in turn responsible for the secretion of the cytokines that in turn activate the TH cells. It's like TH cells activate the macrophages, macrophages in turn activate the TH cells, and TH cells activate the macrophages to the gamma interferons, and the TH cells activate the macrophages activate the TH cells with the help of the cytokines that are produced from them. So like this an interaction exists in between the TH cells and the TH cells and macrophages. Let's move on to the second kind of the cells, second kind of the T lymphocytes are the TC cells. So cytotoxic TC cells are the they are simply can call them as killer cells, and they are provided with a CD8 marker, and they are recognizes the antigen only when they are presented by the APCs, antigen presenting cells. So, once the TC cells comes in contact with the antigen which is presented by the antigen presenting cells with the help of Mhc molecules, then the TC cells get transformed into CTL cell, that is cytotoxic lymphocyte cells, which we call them as effector cells, and which are directly involved in the cytolysis of the infected cells, and there are the altered cells that are present inside the body. So, the secretions of the CTL cells are responsible for the those cells which are actually infected, like especially the viral infected cells inside our body are get killed with the help of the secretions produced by these CTLs. And not only that, if at all any cells that inside our body are get transformed into tumors, those are also get killed by these cells. And so, in that way they are actually involved in the cell mediated immunity. Then let us move on to the third kind of the lymphocytes are large granular lymphocytes, which are called as natural killer cells. And so, they are the most important cells in the entire the defensive system of the body, and they destroy not only the infected cells, but also the altered cells inside our body. And sometimes the viral infected cells are the tumor transformed cells which are present. They are also get recognized and get killed by these large granular lymphocytes. But these NK cells, unlike that of the B lymphocytes and T lymphocytes, they are non-specific, and they can able to destroy the any kind of the infected cells or the altered cells, and especially they are also function without taking the help of the antibodies. That is why their defensive mode of mechanism is without the help of antibodies, that is antibody independent manner they defend against these infected cells or the altered cells. And so, they also can recognize the tumor cells before they become the get activated, and they also become the part and parcel of the innate immunity, and especially showing the defense against the infected cells and the tumor cells. And another kind of the cells that are present in our body are the in the immune system, cells of the immune system are the phagocytes. And phagocytes means the cells which are responsible for the engulfing. And so, those are the two different types are present, and they are classified into two different types. One is MNPs and another one is PMNs. So, mononuclear phagocytes and the polymorphonuclear phagocytes, like that there are the two different types are present. And mononuclear phagocytes, they are called MNPs. So, as long as you know that WBC cells are of the two types, and granulocytes and agranulocytes. Under the agranulocytes you have got the lymphocytes and the monocytes. So, as long as they are freely circulating inside the blood, and we call them as monocytes, sometimes these monocytes come out from this blood circulation or from the blood capillaries, enter into the surrounding tissues, and that is what we call it as entering into the extra capillary spaces. And so, where they get transformed into the various forms of these macrophages. And so, depending on the kind of the tissue into which these monocytes has moved into, one like the extravascular or extra capillary spaces. So, based on that they have given the names. And for example, if they get settled in the connective tissues, we call with the name histiocytes. And if they get entered into the liver, we call with the name Kupffer cells. And if they entered into the brain, then we call them as microglial cells. If they entered into the bone, and where we call them as osteoclasts, and in synovial fluid we call them as a synovial cells. And so, these are all nothing but the monocytes, and there are also we can call them as resting macrophages, and these cells only get activated and those respective organs are get infected, and as a result of infection only all these kinds of the cells become again activated and helps in the defending the body against those infected agents. Then move on to the another kind of the phagocytes are polymorphonuclear phagocytes, and why the name poly, many, morpho, shapes, nucleus. And so, nucleus acquires the different shape like bilobed or trilobed or the many lobed, and like that inside that nucleus acquires the different shapes. So, we call them as polymorphonuclear leukocytes, and which are nothing but the granulocytes as we studied in previous classes. And so, these are neutrophils, basophils and eosinophils. And now, these are the mainly the out of all these eosinophils and the basophils and neutrophils. Neutrophils are actually the major cells which are actually major phagocytic cells out of these all the three, and in which the nucleus is actually exist in the 3 to 5 lobes, and so they are the major phagocytic cells compared to that of the basophils and eosinophils. Coming to the auxiliary cells, and so these are the cells, though they are not directly involved in the immunological responses, but they help the immune cells of the immune system. And so they include actually the basophils, mast cells, platelets, and the antigen presenting cells. And basophils and mast cells, they are responsible for the production of or the release of inflammatory mediators, and like heparin or the histamine, they are actually released and which are responsible for the production of the inflammations. And so inflammation is also a part of the second line defense. And then antigen presenting cells, and because especially the T lymphocyte cells, TH and TC cells cannot able to recognize the antigen, no. So unless they are presented by the antigen presenting cells with the help of MHCs, so they cannot able to recognize the antigens, and so we need the antigen presenting cells. And so which cells of our body are the immune system actually functions as antigen presenting cells, and these are, one is the macrophages, a large phagocytic cells. Of course, B cells also can able to function as antigen presenting cells, but out of all these the most potent one is the dendritic cells. And so dendritic cell is the one in which the provided with the cytoplasmic processes are present, highly branched cytoplasmic processes are present, which are called almost look like similar to the dendrons of neurons, so we call with the name dendritic cells. And so antigen presenting cells are responsible for the presentation of these antigens to the TH cells, and so and which are get produced with the help of only Mhc molecules and major histocompatibility molecules. And now move on to the another topic under this immune system is the organs of the immune system. And so what are these organs? And they are involved in the organs of the immune system. They are responsible for the origin of the immune cells, and they are also responsible for the maturation of these immune cells, and they are also responsible for the making these immune cells to get proliferate. So one is the origin, that is a production, and make them to get proliferate, increase in their number, and also make them to get differentiated. So all these are done with the help of the lymphoid organs. And so they are divided into two different types, and one is the primary lymphoid organs and the secondary lymphoid organs. And so primary lymphoid organs are those which are responsible for the production, and after their production these organs are also responsible for making them to mature and make them to become immunocompetent, so that they can, immunocompetent means so that they can able to recognize the antigen, and antigen recognition capacity is attained by these cells and under the influence of the primary lymphoid organs. And examples are the bursa fabricius, it is a lymphoid organ and located near to the rectum in case of the birds, and whereas in our body the bone marrow and the thymus gland in mammals. And so these two actually functions as the primary lymphoid organs. And now second one is the secondary lymphoid organs, and secondary lymphoid organs are those which are responsible for making these mature cells of the immune system to become functional lymphocytes in these organs. And so these are the organs which are responsible for the providing the sites for interaction between the antigen and the immune cells. And so as a result of that they become functional cells, especially they transform into effector cells. And so they provide the sites for the interaction between the antigen and the immune cells. And so in that way then after that interaction is over, these are the organs, that is secondary lymphoid organs, in which the cells get transforms into functional cells, and especially the effector cells. And examples for these secondary lymphoid organs are spleen, lymph nodes, tonsils, Peyer's patches of small intestine, and the appendix and worm-formed appendix. These all forms as an example for the secondary lymphoid organs. Among all them, actually the spleen is the one of the important one, which is the largest lymphoid organ, and as you know that it is a reddish mass-like gland that is located and towards the left side of the abdomen near to that of stomach. And so after left part of the abdomen, it is located just underneath this diaphragm. And so it contains the lymphocytes and also contains the phagocytic cells are present, and one of the major function of this spleen is responsible for the filtration of the blood. And so, at the time of the filtration of the blood, it is responsible for the trapping of blood-borne microbes or the antigens. And so, and it is also responsible for the acting as a largest reservoir for erythrocytes. And after the during the early stages in the embryonic stage, of course, it acts as a hemopoietic, but later part of the life, it acts as a graveyard or burial ground for the old and the worn out of RBC. And so, that is why where it is responsible for the destruction of the age old or the old and worn out RBC cells. And the second kind of the organs are the under the secondary lymphoid organs are the lymph nodes. And so, very small solid structures and that are located all around the body at the different parts of the as a part and parcel of this lymphoid system. And so, they are the small solid structures and which are located at the different parts of the body, especially at the groins, armpits, at the neck region. They all are very much well distributed and which becomes a part and parcel of this immune system. And so, these are also responsible for the trapping of microorganisms and the antigens, and which in turn responsible for the activation of the lymphocytes. And so, almost the 50% of this lymphoid tissue that is present inside our body is actually formed by contributed by the malt and mucosa associated lymphoid tissue, and which is almost present in the entire the gastrointestinal tract and also the respiratory tract, and you will also find it in the urinogenital tracts in which it is presented. So, these are all the different kinds of organs. And the next one we are going to move on to the another kind of the concept related to the immunity is the soluble mediators of immunity. And so, these are the mediators like, and they are responsible for the bringing the interactions in between the cells that are involved in immune system. And so, they are like helps in communication, and especially and that is why they are known as mediators, and which exist in the soluble state within the blood and the various kinds of the body fluids present. And so, they are actually divided into three different types, and one is complement proteins, another one is the cytokines, and the third one is the antibodies. And among them, the first one is complement proteins. And so, these complement proteins and like as you studied earlier in case of the blood clotting, and there are the 13 factors are present which helps in the process of blood clotting, and they are present in soluble or dissolved state within the plasma. In the same manner, even in our blood there are the inactivate protein plasma proteins are present, and they are also forming present on the cell surface proteins. And so, whenever these like there also in the blood clotting you studied, whenever those blood clotting factors are get activated, they become comes into the insoluble manner and helps in the formation of the fibrin. Here also in the same manner, these inactive soluble proteins which are become the part and parcel of the immune system, they are also whenever activated, they become into the they become and form the membrane attack complex, what we call it as MAC. And so, this membrane attacking complex which is formed by these complement proteins in turn responsible for the creation of pores. And we will study how these MAC functions, how the pore is created when we going to study in the mechanism part. And so, this MAC forms the pore in the plasma membrane, allowing the, so whenever they creates the pores, and so whatever the extracellular fluid is present now, those body fluids started entering into the cell and makes the cell to swell and burst, and in that way the complement proteins actually helps. And so, complement proteins are nothing but the inactivate soluble proteins present inside the plasma. Whenever they are get activated, they responsible for formation of membrane attacking complex, which creates the pores on the cell membrane that makes the ECF to get enter inside the cells, so that cells swell and burst, and in that way the infected cells are get killed. So, these complement proteins and can also form a coat over the pathogens and responsible for the attraction of the neutrophils and macrophages, so that these neutrophils and phagocytes can easily destroy those cells. And so, they are also responsible for the attraction of the cells towards the infected cells. And complement proteins and their activities. And so whatever the different kinds of the proteins that are present in plasma and their mode of activity, all together we call it as complement system. And this is one kind of soluble mediators. And next, another kind of the mediators are the cytokines. And cytokines are actually get secreted by the TH cells or the infected cells that are present inside the body. And so they are nothing but the very small soluble mediators are the molecules. And so these cytokines, when they are released into the body fluids and all, so they can able to go and attach to those receptors present on the surface of the cells, and which in turn responsible for the activation of those cells or the differentiation of those cells of the immune system, and which make them to either the phagocytosis process or the cytolysis of the infected cells. And these cytokines are of two types, and one is interleukins, another one is the interferons. So interleukins, which are actually produced by the leukocytes, which are primarily involved in the differentiation of the immune cells. And see, suppose if the lymphocyte has to get differentiated into the B lymphocyte cell, B lymphocyte cell has to differentiate into the plasma and memory. This differentiation is actually under the control of these cytokines. Or the T lymphocyte cell has to differentiate into the TH and TC cell, and the TH in turn should differentiate into functional TH cells, and the TC cell to differentiate into CTL and memory cells. So this becoming into CTL and memory cells, that is what we call differentiation, so which is actually under the control of these cytokines. And so they are the functions of that cytokines. And next, another kind of the cytokines are interferons. They are very, very important cytokines. And so they are actually produced by an antiviral, they are the antiviral proteins which are produced by the viral infected cells, and so to protect the neighboring cells. And so it is like a way of communicating with the neighboring cell, and already the cell which is infected with a virus, it is passing that information to the neighboring cells to develop the antiviral state, and so that the virus cannot able to get attack the neighboring cells. And so this which is actually produced by the infected cells itself. And so like that we have got the three different types of interferons are present, and alpha, beta and gamma interferons. About the gamma interferons we already studied. TH cells are responsible for production of gamma interferons, which stimulate the phagocyte cells. Phagocyte cells produce macrophages. Macrophages in turn produces the cytokines. Cytokines in turn activate the TH cells. There we studied about the gamma interferons. And then the third kind of the soluble mediators are the antibodies. And antibodies are actually produced by the B lymphocyte cells, and these antibodies are very highly specific. And so they antigen and antibody, these two interactions are very specific. And so in these antibodies, the part or the portion of an antibody with which we can able to recognize the antigen, which we call it as paratope. And so antibodies are freely circulating inside the body fluids, and sometimes these antibodies get classified into two different types. And some antibodies which are actually get attached to the surface of the B cells, we call them as membrane bound antibodies. The antibodies that are produced by the plasma cells and freely moving within the body fluids, we call them as the freely circulating antibodies. Like that, the two different types of the antibodies are present. Now Rodney Porter is the scientist who actually proposed the structure of the antibody. So the antibody has the structure which is proposed by this particular scientist. So the antibody almost gives appearance of a Y-shaped molecule, and if you look at the structure of this one, the antibody appears in the form of a Y-shaped structure, in which there are the two polypeptide chains are present, and which are called as two polypeptide chains, and two heavy chains and two light chains are present, and which are named as H2 and L2, which are present. Now these two chains are held together at the center by means of the disulfide bridges are present. So with the help of these disulfide bridges, the two heavy chains are attached to each other. Light chain and heavy chains are also get attached together by means of this disulfide bonds. Now this antibody which all these light chains and the heavy chains, which are all are arranged in the form of a Y-shaped molecule. No, in this that the two arms are present and the one stem lichen which is present, which has got the two ends now on. So the ends of the arms are called as antigen binding sites, and these are the ends where the antigen comes and the binds, and so that's why they are called as antigen binding sites, and this is an antigen. So the portion of the antibody which is responsible for the binding with the antigen, so the part or the portion of the antibody which binds with an antigen is called as paratope. So paratope is nothing but antigen binding site, antigen binding site. So which are present at the ends. So this depending upon the sometimes the same type of antigen binding sites or the different types of the antigen binding sites are present. So that is a fragment which is responsible for binding of antigen. That's why it is called fragment antigen binding site, FAB end, and we can call it as, that is the ends of the arms of this Y molecule or the antibody. Then comes to the opposite to that one, the other end, and where we call it as, this is the end, we call it as FC end. FC means it is a crystallizable or the fragment crystallizable or the fragment cell binding end. So this is the part with which it gets binds with the cells or the crystallizable end. So in this there are the another certain regions that are present are in this Y molecule. So from here to here, the whole region that is present in these parts of the any antibody, they are regarded as variable regions. And so depending upon the different types of the antibodies, these are the portions which may change in their particular structure. But there is another part which is present from here to here, so which is called as constant regions. So the constant region, that is for all the different types of the antibodies, these portions will remain constant and they do not undergo any change. Only the ends of the arms of this Y molecule or the antibodies, they keep on changing in the different types of antibody. In that way, they are called as variable regions and the constant regions. So both variable and the constant regions are provided with light chains as well as heavy chains, and both are present in this antibody molecule. So now based on the structure of these antibodies, and the antibodies are get classified into different types, and so there are the different types of antibodies are present, so they are classified into different ways. And so they are actually classified into different ways. They are like IZ, IgE, IgD, IgA or IgM. As you know that antibodies are also called as immunoglobulins, and these immunoglobulins depending upon their molecular weights and the shapes and the other structures, they have given the different names, and ADE, GM, like alpha, delta, giga, mega and the epsilon, and giga, epsilon, delta, alpha, mega, like that they have given the different names depending upon their structure, molecular weight and the and of their structures. They are divided into the 5 different types, and so all of them actually fall into these, any kind of the antibody, all of them falls into these 5 different kinds of the antibodies only. And now among them again they are divided into the different types, and like IgE, E, D and G, they are all classified as monomeric type, and that means they are only the whatever the structure or the shape that we studied now, in that manner they are presented. Whereas IgA is a dimeric type, and that means they are actually 2 different kinds of the antibodies with their Fc ends bind together and show the response. And whereas IgM is the pentameric type, and now all of them can bind with each other to form a pentameric form. And so there are the structure depending on that, M is functions as a pentameric one, A is the dimeric form, whereas the G, E, D are of the monomeric type. And so the manner in which they mount the attack and their structures, they are also divided into the 3 different types, and monomeric, dimeric and pentameric. Now let's move on to the another one or the part of this immune system, that is the antigens. So these are the substances which can able to induce the detectable immunological responses are called as antigens. Like we can also define like any foreign substance that enters inside the body, but every foreign substance cannot able to generate the immunological response inside the body. Unless our body recognizes it and shows the immunological response, we cannot regard it as an antigen. So the foreign substance that enters inside the body and induce the detectable immunological responses, we call it as antigens. So these antigens can be of any different kinds of the substance. They can be of the proteins, or they can be of the polysaccharides, or the nucleoproteins, nucleic acids. They can be of the any different types. So these antigens are the freely circulating antigens if they are present, which can be easily recognized with the help of a B cell receptor located on the B lymphocyte cells. Whereas as you studied earlier, the T lymphocytes can also recognize the antigen, but they has to be presented by the either antigen presenting cells or the altered cells. Now there are the certain parts of the for example, if you take an antigen, this is a particular antigen on which the provided with then a surface which is actually recognizable with the help of an antibody. So the part of the antibody which attaches to the antigen, as we studied already, is called paratope. Now the part of the antigen which is recognized by the antibody, we call it as epitope. So as antigen can have the same kind of the epitopes, or the different kinds of the epitopes are also present. And based on their existence, these antigens are also get classified into different types, and freely circulating antigens that are circulating inside the body, or sometimes the antigens that are produced within the infected cells, and they are called intracellular antigens. For example, if the virus attacks the cells, and those antigens are get produced inside the cells only. Sometimes the antigens that are present on the surface of the cells, and like for example blood groups, as you studied now, the A antigen or B antigen or RH, they all are located on the surface of the cells. And so antigens present on the surface of the cells, antigens that are produced by the infected cells within the cell present, they are intracellular, and antigens that are freely circulating inside the body fluids. And now once these antigens enters inside the body, we need to process these antigens to make it recognizable by the TH cells and TC cells especially. And so for that purpose, we need the processing of the antigen. And processing of antigen is nothing but the removal of all the unwanted surfaces or the unwanted content that is present inside the antigen. So only the part or the portion of the antigen that can able to induce a detectable immunological response. So that phenomenon we call it as the processing. So whenever any antigen enters inside the body, and that antigen is engulfed by the phagocytic cells, and after that engulfing process is over, the lysozymes that are present inside the cell is responsible for the removal of all that unwanted regions and release only the antigenic polypeptides. So those determinants, which are nothing but the epitopes. So once that process is over, then that is get presented to the outer surface of the cell. For example, this is an antigen that enters inside the body, and which is a phagocytic cell. This is a simple phagocytic cell. So phagocytic cell is responsible for the production of these pseudopodia and trying to engulf this antigen. And now they call it as antigen, and which is a phagocyte or a macrophage. So this process we call it as engulfing. So as it is engulfing the antigen, at the later next stage it enters into the inside the cell. So once this entire the process is get completed, now the antigen comes to the inside the cell. So this the process of the engulfing or the phagocytosis is over. Now inside the cell, the lysozyme is present, which is provided with the various kinds of the enzymes, and all the hydrolytic enzymes are present, which comes in contact with this phagosome. Now, the interaction of the lysosome and the phagosome releases all these enzymes and responsible for the processing of this antigen. So, that means it is responsible for the removal during this processing stage. All the unwanted structures or the elements that are present in the antigen are get digested, are removed, and only releasing only the antigenic polypeptides into the outside the cell. So, only antigenic polypeptides comes out and they are liberated into the cytoplasm, so which are called as antigenic polypeptide. So, this step we call it as processing. So, once the processing step is over, then the next step that comes to role played by these phagocytic cells is the presentation. So, presentation is the presentation of these antigens. So, here the role of actually the Mhc molecules comes to the way, that is major histocompatibility molecules. So, whatever these antigenic determinants, so these we call it as antigenic determinants. So, these antigenic determinants that are liberated inside the cytoplasm of the cells, which are made to present on the surface of the cell with the help of the molecules, and they are actually called as Mhc molecules. So, Mhc molecules are these are the specialized molecules which are present inside the cell, which are present inside the cell only, and which are produced from the nucleus of these cells. So, these Mhc molecules, while they are coming out from inside the cell to the outside the cell, they interact with these antigenic determinants and bring to the surface of the cell. So, these are called as a major histocompatibility molecule. The function of the major histocompatibility molecules is getting interacted with the antigen inside the cytoplasm of antigen presenting cells and make it to bring to the surface of the cell, so that they are made to available to the TCR of TH cells and TC cells, so that they can easily recognize the antigen. So, that is actually the function of these Mhc molecules, and which are of two types. Mhc molecules are again divided into the two different types, which are the class 1 and class 2. Like this, there are the two different types of the cells are present. Now, Mhc class 1 molecules are present in all the nucleated cells. And so, any cell which is provided with a nucleus, suppose almost all the, like except the RBC or some other cell, almost all the cells present inside our body are provided with a nucleus, which are provided with the class 1. Whereas they present the antigens, and so of course any Mhc molecule, whether class 1 or class 2, they present the antigen, but the antigen that is presented by the class 1 is recognized by the TC cells, which are TC cells, and so antigens of the TC cells are present these antigen, and present the cell surface to the APs and lymphocytes innate to the class 1 molecules. And now they present the antigens to the also to the TH cells for the cell mediated or the humoral immunity, and T cells have antigenic specific receptors are present on its surface, so that they can be easily recognized by the antigenic bounded molecules to the Mhc molecules. And so, with this another kind of the another one which we have got is the class 2 one. So, we will write a small step, so that you will understand this Mhc molecules. And for example, an antigen presenting cell responsible for the presentation of an antigen with the help of the class 2 one. With the help of class 2 Mhc molecule presents that antigen. So, antigen which is presented by the class 2 Mhc molecules by the antigen presenting cell, which is recognized with the help of TCR or ASR, antigenic specific receptor or T cell receptor, of those cells which are provided with a CD4 marker are called as TH cells. So with this single step or the equation we can easily recognize, and see for example, antigen presenting cells are responsible for the presentation of the antigenic determinants with the help of MHC class 2 recognized by the TCR or the ASR, antigen specific receptors of those cells that are provided with a CD4 marker are called TH cells, or which are also called the T helper cells. And now in reverse to that one, altered self cells. And so altered self cells are provided with the MHC class 1, which are also responsible for the presentation of antigen, which is also made recognized with the help of the TCR or the ASR, that is T cell receptor or antigenic specific receptor, which is made recognizable by those cells which are provided with a CD8 marker are called T C cells, and that is T cytotoxic cells. And so the basic entire the immunological function is present in these two equations, and so don't get confused with this. CD4 marker is present on the TH cells, CD8 marker is present on the TC cells. Both are responsible for the recognition of antigen only when they are presented by the either antigen presenting cells or altered self cells. Altered self cells are nothing but the infected cells that are present inside our body. And under the types of immunity, based on the nature of response, immunity is actually get classified into two different types. And so in what way we are showing the response, and one is innate immunity and another one is the adaptive or the acquired immunity. And so in case of this innate immunity, it is an inborn resistance and present inside the body, and so it is present inside the body from the time of the birth. And this kind of the immunity is almost common to the almost all the kinds of the living organisms, and it is actually non-specific, which means that it can able to show the defense to the any kind of the microbes, and it won't show the any kind of the partiality like, or it can able to offer the resistance to any kind of the body. And it already exists as inside the body, so it do not require any prior contact with the microorganisms, and also it does not provide any kind of immunological memory. And so but it is actually provided with the various different kinds of the barriers are present, and it is actually that this kind of immunity is executed by different kinds of the barriers. And so among them, the first barrier is actually the physical barrier, and physical barrier means the skin, mucous membranes that are present part and parcel of this respiratory and gastrointestinal tract or the urinogenital tract. They are all actually acts as a physical barriers. And of course the skin and the microbes are the very potent physical barriers, and which can able to prevent the entry of those microbes. And the second kind of the barriers that comes under this innate immunity is the physiological barriers. And physiological means as a result of the metabolism inside the body, we do secrete the certain secretions. For example, inside the stomach, the gastric gland secretes the HCl, and which is highly acidic in nature, and where it is most of the microbes are get killed. And another one is the lysozyme that is present inside the saliva, lysozyme that is present inside the tears. They also potently functions as the physiological barriers. And in addition to that, the sweat glands produces the sweat, and the sebaceous glands that produces the sebum, their pH is actually different. And so at that pH, whose range is actually at about the 3 to 5, certain kinds of the microbes cannot able to survive. And so they all are responsible for the inhibiting the growth of the microbes. And so in that way they provide the one kind of the barrier. And the third kind of the barrier is the cellular barriers. And so after crossing all these, now enters inside the body. Once these microbes enters inside the body, then the role of the phagocytic cells will come on, like the neutrophils or the natural killer cells, monocytes. All the different kinds of the cells are present, and so they are actually responsible for the showing the defense, and which comes under the part of this cellular barriers. And then the fourth kind of the barrier is the cytokines, and just now we studied under the soluble mediators, and they are the molecules that are released by the immune cells of our system, or the another kind of the molecules are produced by the cytokines are produced by the viral infected cells. And so which are responsible for the, one is the proliferation and another one is the differentiation, and for both these the cytokines are actually responsible, and so they protect the non-infected cells from the further infections. And so all these are become the part and parcel of this innate immune system.